Okay, hello everyone. So let's go ahead and get started with chapter 4. Chapter 4 is all about eukaryotic cells. So in this chapter, we're going to talk about several different categories of eukaryotic cells. We will start by talking about the endosymbiotic theory, which is the theory of the origin of eukaryotic cells. Then we will move on and we will talk about fungi, very briefly about algae. We will move on and talk about the protozoas and then the helminths, those parasitic worms that we talked about in chapter 1. We will talk about vectors and what vectors are, what they do, why they are related to some of the parasites and what they do for us. All right, so if you remember, we talked about the three domains back in chapter 1. And I said that the three domains are bacteria, archaea, and eukarya. I mentioned that the group the archaea didn't exist until 1978. It was thought that the archaea were bacteria because they're shaped like bacteria, they behave like bacteria, they're very much similar or have a lot of similarities with bacteria. But if you look at how they actually branch off of the eukarya because based on genetic sequence, archaea may look like bacteria, but they're more closely related to us. Okay. But now if we look at the far left, let's look at the domain bacteria. So at the very bottom, you see the first branch is Thermatoga. They estimate that the world is approximately 3.5 billion years old. And they think that Thermatoga was among the very, very first organisms. Therm meaning heat. They thought that the world was a much hotter environment during that time period, and so that's how they came up with Thermatoga. Now, branching off from that are your gram-positive bacteria, and then later on came the gram-negative bacteria. They think that the gram-negative bacteria evolved after the gram-positive bacteria, and that's because the gram-positive bacteria only have one plasma membrane or one cell membrane, whereas the gram-negative bacteria have two cell membranes. So they thought that the gram-negative bacteria are more evolutionarily advanced than the gram-positive bacteria. Now, one thing you also might notice, under bacteria, you see mitochondria and chloroplast. That's right. It was thought that at one time, mitochondria and chloroplasts were actually bacteria. Now, today we know them as organelles of eukaryotic cells, but that wasn't always the case. And on the next slide, we're going to talk about the endosymbiotic theory, or maybe 2 slides from now, we'll talk about the endosymbiotic theory and talk about how the mitochondria and the chloroplast went from free-living bacteria to organelles in eukaryotic cells. And then if you look over at the domain eukarya, you see the archozoas at the very, very bottom. We'll talk more about those later today in this chapter. Microspora, which we'll also talk about in this chapter. And then all the way up to the fungi, the animals, and the plants. So pretty much everything within the 3-domain system is microscopic, with the exception of fungi, animals, and plants. Okay, so there is one takeaway point from this slide. This slide is just review, except for one point, and that is the scientist Carl Weiss, and the year may look familiar to you, 1978. It was Carl Weiss who first did genetic sequencing on that group of organisms that were thought to be bacteria, and it was found out that they were actually archaea. So Carl Weiss was the person that discovered the grouping, the archaea, and gave them their grouping, gave them their heading, archaea. Okay, so now we are up to the endosymbiotic theory. This is a theory that attempts to explain the origin of eukaryotic cells. Let me start off by saying this is just a theory. A theory is something that cannot be proven. They use data to come up with a best guess type situation, but they can't actually prove this. So there are several different theories as to how the origin of eukaryotes came to be. But this is the most commonly accepted theory as to the origin of eukaryotes. So let me just read through what I have here on the slide, and then I'll try to put it in my own words and kind of explain it to you briefly. So the endosymbiotic theory is an evolutionary theory that explains the origin of eukaryotic cells from prokaryotes. It states that several key organelles of eukaryotes originated as symbiosis between separate single-celled organisms. According to this theory, mitochondria and chloroplast and possibly other organelles representing formerly free-living bacteria were taken inside another cell as an endosymbiont around 1.5 billion years ago. Okay, so symbiosis. We talked about symbiosis the other day when we were talking about relationships. So all of symbiosis is, that just means that there was some type of relationship between two different organisms. Okay. So basically, if I were to sum up the endosymbiotic theory, it basically states that there was some organism 1.5 billion years ago that took up mitochondria and chloroplast. And with this new relationship, both parties must have been happy with this new living relationship. The host cell was happy. The mitochondria and the chloroplast were also happy with this new arrangement. So at that point, every time the host cell reproduced, the mitochondria and the chloroplast also reproduced. So now every time that host cell was found, it also had mitochondria and chloroplast within that cell. And so that's how the origin of eukaryotes is thought to have occurred. It started off with the uptaking of smaller, simpler molecules into something that's larger. Now, there is what they call a modern day example of endosymbiosis, and it's with the organism Cyanophora paradoxa. So paradox is Latin for puzzle, because scientists are kind of puzzled as to how this living arrangement came to be. Every time they find Cyanophora paradoxa, which is a protozoa, one of those single-celled animals, every time they find this protist, they find a very particular bacterium that lives inside the protozoan, and it takes up the majority of the space of the protozoan. You can see a picture of that here. So they consider this a modern day example of endosymbiosis. And so basically their thought process is, if it happened with Cyanophora paradoxa, who's to say it didn't happen a billion years ago with the mitochondria and the chloroplast? So as far as evidence that supports the theory of the endosymbiosis, here's evidence. Both mitochondria and chloroplast have their own DNA. No other organelles inside a eukaryotic cell has their own DNA. Okay? But mitochondria and chloroplast do. They have their own DNA. Second, they have 70 S ribosomes. Remember, we mentioned ribosomes in the past. Bacteria or the prokaryotes tend to have 70 S ribosomes, whereas eukaryotic cells tend to have 80 S ribosomes. Well, the mitochondria and the chloroplast have 70 S ribosomes, which is similar to that found in bacteria. Next, the mitochondria and the chloroplast have double membranes. All other organelles only have a single membrane. This is important because during the uptaking process a billion years ago, it was thought that part of the host cell membrane was pinched off. And so that is how the mitochondria and the chloroplast wound up with a second membrane. Okay? Again, they're similar in size to bacteria. They replicate by a process that's very similar to that of bacteria, which is binary fission. Also, they have genes that resemble certain bacterial genes. So this is all evidence that supports the theory of endosymbiosis. There's a more recent piece of evidence that's not even on this list yet, but there have been some recent studies performed where they have actually removed mitochondria and chloroplast from living cells, and the mitochondria and chloroplast will continue to survive on their own, free, out in the open without being attached to some type of host cell. So again, that is some newer evidence that supports the endosymbiotic theory. All right, so the first topic that we are going to cover in the eukaryotes are the fungi. Keep in mind, we are in the eukaryotic chapter. These are eukaryotes. They are going to have a nucleus. They are going to have, for the most part, they are going to have traditional organelles, like you would see with a traditional eukaryotic cell, like you studied in A&P, like a human cell. So there's a lot of similarities here because we are not talking about prokaryotes in this chapter. We are talking about the eukaryotes, and a lot of these are going to be very similar to human cells. All right, so the first term you see under fungi is you see the term chemoheterotroph. This is a long word. Learn it. You will see this at least 4 other times between now and the end of the semester. So a chemoheterotroph is any organism that uses carbon as a food source and as an energy source. Okay. All right, so fungi fit that definition. They are chemoheterotrophs. They use carbon as a food source and as an energy source. Well, as it turns out, humans, we are also chemoheterotrophs. Most organisms are chemoheterotrophs. So if I were to ask you, give me an example of something that's not a chemoheterotroph, well, the opposite of a chemoheterotroph is something known as a photoautotroph. Any organism that can perform photosynthesis is not a chemoheterotroph. So again, most living organisms that breathe oxygen, that eat or ingest some type of food particle, are going to be chemoheterotrophs. Now, some of these chemoheterotrophs are aerobic, such as the molds. That just simply means aerobe means they need oxygen in order to survive. We will also talk about the yeast, which are facultative anaerobes. So this is a new term. A facultative anaerobe is an organism that grows better if oxygen is present. However, if you remove oxygen from the environment, they still grow, just not as well. Okay, so that is what a facultative anaerobe is. And learn it, don't forget it, because when we talk about bacteria in later chapters, it turns out that some bacteria are also facultative anaerobes. All right, so mycology, the prefix myco means fungi. So mycology, this is simply the study of fungi. Most fungi act as decomposers. Again, we learned this in chapter 1. The main job of fungi is they decompose organic matter. So the example I gave previously were things like tree limbs or leaves or grass. Once it dies, it eventually is broken down. Well, what breaks it down? Fungi. One of the key roles or the main roles of fungi is to break down or decompose organic matter. There are only a few that are considered to be parasites of plants or animals. And I'm really only going to focus on the ones that tend to infect humans, okay? But there are definitely fungal infections that can infect plants as well. For example, wheat rust is a fungal infection that infects our wheat crops, okay? But other than that, we'll go back and let's talk about some of the common fungal infections that we see in humans. The first one, for example, you see the foot, and then you see at the bottom the toenails. Well, that is because athlete's foot is a very common fungal infection. The scientific name for athlete's foot is Tinea pedis. Pedis, again, means foot. So Tinea pedis, again, athlete's foot, is a very common fungal infection. It is highly contagious. Okay. If you've ever lived in a dorm style situation where you share a shower with many other people, a lot of times they will tell you to wear shower shoes because this is a fungus that likes warm, moist environments. And that's the perfect breeding grounds for this fungus. Well, what is a shower? It's typically warm and moist. Okay. Men tend to get athlete's foot at a higher rate than women do. And again, it goes back to the jobs that men tend to have when compared to women. A lot of men tend to work outside. They wear work boots. Their feet get hot and sweaty. And that is the perfect breeding ground for this particular fungus. As far as treatment, so athlete's foot is not fatal. I have never heard of a person dying from athlete's foot. It's one of those that is characterized by an extreme itch, redness of the skin, sometimes yellowing of the skin, peeling of the skin, a yellowing of the nail beds. It's a very uncomfortable condition. Again, the main sign and symptom is an extreme itch. Okay. As far as treatment is concerned, yes, you can go to your local pharmacy and buy over-the-counter treatments, but the problem with those is they are becoming ineffective. And that's because the fungus has been exposed to these medicines so much that the fungus has mutated to the form or to the point that typical over-the-counter antifungals are no longer effective for things like athlete's foot. So a lot of times you still have to take a trip to the doctors and get a prescription-based treatment for something like athlete's foot. Another example of a fungal infection, if you look at the very, very top center, you see this circular fungal infection. That is ringworm. All right, so ringworm is not a worm. It is a fungus. Okay. Again, highly contagious, and it typically appears on the extremities, so on the arms and legs. Because what happens is a person may have ringworm, and sometimes they don't realize it, but it's itchy, and they can scratch it. And what happens when you scratch is you get the fungal spores, which we'll talk about in a minute. You'll get the fungal spores stuck under your fingernails. And then you go and you touch another person, and you pass those fungal spores on to another person, and they wind up contracting ringworm. Also, cats are one of the main carriers of ringworm. So if you see a stray cat and you attempt to pet the stray cat, if they have ringworm, it's very possible that you will contract ringworm. Again, it's not deadly. I've never heard of a person dying from it. It's one of those, though, it is highly contagious, and it's more irritating than anything. Okay? It causes redness. The real risk here is if you're scratching it, at some point you may wind up opening the skin and creating an open wound. At that point, it could potentially become more serious because now bacteria from the air, from the environment, could potentially enter that open wound. So at that point, it could potentially become more serious. But from the ringworm itself, it's not deadly. It doesn't really cause major illness. Okay, now next you may see a picture of a tongue, and that tongue is coated in white. That is called thrush. It's very, very common in babies. It's also common in the elderly because as people become older, their immune system is naturally suppressed from age, allowing the yeast, and the scientific name for this yeast is Candida albicans. Candida albicans. This is the yeast that causes thrush in mouths. Now, as far as females are concerned, females sometimes wind up contracting yeast infections. That is another fairly common fungal infection. Okay, but trust me, I spared you. I did not show a picture of this. Okay. But the interesting thing about yeast infections is that it's also caused by Candida albicans. It is literally the same yeast that causes thrush in mouths. It's just a different location. So a yeast infection in women is called candidiasis. So again, the scientific name for a yeast infection is candidiasis. Okay, so I think I've covered all of the points that I want to make as far as some of the more common fungal infections. Now, it says the number of serious fungal infections are increasing. There is one in particular. The scientific name for this fungus at one point in time was called Pneumocystis pneumonia. They have since renamed it. It has now been renamed Pneumocystis jivency, but it causes a fungal type of pneumonia. However, it really only infects those that are immunocompromised. So much to the point that really the biggest risk is with HIV patients. Because when HIV, after a person has had HIV, what HIV does is it suppresses the immune system. So they have seen a very strong correlation between HIV and pneumocystis pneumonia, this fungal type of pneumonia. The thing about pneumocystis pneumonia is it's oftentimes fatal because it only infects those that have a severely compromised immune system. We don't have a lot of great antifungals on the market. There has not been a lot of money or research put into new antifungals. So for that reason, when a person contracts pneumocystis pneumonia, a lot of times that person will die from pneumocystis pneumonia because it means that they have a severely weakened immune system, and coupled with the fact that we don't have great antifungals. Okay, now let's move to the very, very bottom of this slide and let's talk about hyphae and spores. The first thing I want to point out, let's talk about spores. Don't confuse an endospore with a fungal spore. They are not the same. They are completely different. We talked about endospores in the past couple of chapters because we mentioned that there were a couple of species of bacteria that produce endospores. Well, when it comes to fungal spores, there are a lot of fungal species that produce fungal spores. You can see in purple, that is a picture of a fungal spore. They are microscopic. They are very, very lightweight, and they spread very easily. An example of this, if you've ever seen a mushroom growing in your yard and you stomped on the mushroom or kicked the mushroom, a couple days later you may see a whole uprising of mushrooms. It's because when you kicked the original mushroom a few days ago, you spread fungal spores, and those mushroom spores spread, they landed, and they started growing. So it's very easy for an animal outside to pass or transfer fungal spores from one location to another, or a bug, or if wind blows. There's a lot of ways of transferring fungal spores, again, because they're so lightweight. Now, the spore will germinate, and you can see that a hyphae will come out of the spore. That hyphae is pictured in white, and again, we talked about hyphae back in chapter 1 when I talked about mycelia and the hyphae, which are the two main parts of molds. So the hyphae come in two forms. They come in what's known as the septate hyphae, and they also come in the term is called coencytic, but I typically tend to call them non-septate hyphae, and your textbook calls them non-septate hyphae. So remember that fungi, these are eukaryotes, so there is going to be a nucleus. So if you look at the septate hyphae, these have septums. It almost looks like cell walls that separate them, and you can see that each area has a nucleus because these can get quite large. These can be macroscopic, and so they are composed of numerous partitions, and therefore each area, each section will have a nucleus. Now, if you look at the non-septate hyphae, It does not have septums that separate it into different sections, but you still see it has numerous nuclei. Okay, so those are two different forms of molds or of hyphae that you can find, and these hyphae are found on molds. If you've ever bought bread and left the bread out for too long, you eventually will see a mold, and that is because molds, for the most part, they are able to use the same nutrients that we do. So that mold landed on your bread, and it's been using that bread as a food source, and it's been growing. And so at some point that mold will get large enough that you can actually see it. And so typically you can see the center mass of that mold, which is the mycelia, and you can also see the fine hair-like projections, which are the hyphae. All right. Okay, so moving on and talking about some more characteristics of fungi. The top couple of terms I just talked about, the hyphae and the mycelia. Molds, they are mostly filamentous. Again, those hyphae, they have those hyphae, which are those hair-like projections. Yeast are typically non-filamentous, and they are typically unicellular, so they're composed of a single cell. All right, let's talk about yeast and how some yeast divide. So we know that bacteria divide through binary fission. We talked about how molds divide on the previous slide with their hyphae and their spores. Well, now when it comes to yeast, yeast typically reproduce through a process known as budding. Okay, so in the upper right hand corner, that is a process of the budding process. It starts off where you have a parent cell, and that parent cell will have a small protrusion that starts to grow out from it. And that protrusion will grow larger and larger until it's about the same size as a parent cell. And then it gets cut or cleaved, leaving behind a bud scar. Okay, so you can see in that picture there is a parent cell and there are several bud scars left behind. Now, budding is also a form of asexual reproduction. So there was no exchange of genetic material. All of those yeast cells should be identical to the parent unless mutation occurred. Dimorphic fungi. These are very interesting. It turns out that most of the fungi that are dimorphic are also human pathogens or pathogenic. These are fungi that will appear more like a yeast at 37 degrees Celsius. And so that is roughly 98.6 degrees Fahrenheit human body temperature. And they appear more like molds at 25 degrees Celsius, which is more like room temperature. So you can have a fungus grow more like a yeast or a mold depending on what temperature it's grown at. So again, that's something that I find very, very unique or very, very different. The next bullet are just talking about fungal spores. Just kind of to recap, don't confuse fungal spores with bacterial endospores. They are completely different. Most fungi tend to grow best in a slightly acidic environment. They typically grow best at a pH of around 5, whereas neutral is about 7. They tend to grow best in low moisture, high osmotic pressure environments, but that's not always the case because I just said a few minutes ago that some fungi tend to grow best in very moist, warm environments, places like the shower. Okay. Fungi, remember, they act as decomposers. So they're able to metabolize complex carbohydrates such as lignin. So lignin is found in tree trunks. Okay. And they're one of the few organisms that have the ability to break down and metabolize some of these complex carbohydrates. So not all fungi are bad. Here's a list, first of all, starting off. Over 600,000 different species of fungi have been isolated and identified. We've talked previously about just a handful of pathogenic fungi, but now here's some fungi that are actually very beneficial to us. There's a fungi we talked about in chapter 1 that is not even on this list, and that is Penicillium, the fungus that we get the drug penicillin from. So it saved thousands, if not more than thousands, hundreds of thousands of people by now. Moving on to Saccharomyces cerevisiae, we talked about this. This is baker's yeast. This is why bread rises. It's also used in wine fermentation or the production of wine. It's also used to grow certain vaccines or certain viruses that are used for certain vaccines. When we get to the virus chapter, I will talk about viral replication and how we grow viruses, why it's important to be able to grow viruses. Some viruses can be grown in cell culture. And so Saccharomyces is a very good choice for growing certain viruses. And so we'll talk more about that. Trichoderma produces a large amount of cellulase. So cellulase is an enzyme that breaks down cellulose. Humans, most animals lack cellulase. That's why we cannot consume cellulose. We can't break it down. Okay. Certain fungi, certain insects such as termites, they do contain the enzyme cellulase. Well, as it turns out, Trichoderma is a fungus that produces a large amount of cellulase, large enough that it makes sense for companies to be able to extract cellulase from Trichoderma and sell it for research purposes or be able to sell commercially to the public. Next is Taxomyces. There's an anti-cancer drug known as Taxol. And I did a little research on Taxol. It's available as a pill or as an injectable. It was actually accidentally discovered, so the history of Taxol and how that came to be is kind of interesting. Next is Entomophaga, which is used for the biocontrol of pests such as mice and rats. And then Paecilomyces is a fungus that kills termites. So those are used a lot in the biocontrol of different types of bugs and insects. Fungal diseases, these are known as mycoses. Okay. So there are systemic mycoses. Systemic means that it infects the entire system. So infected deep within body, affects many tissues and organs. A lot of times these tend to be very, very serious because, again, due to the lack of antifungals that are available on the market. Histoplasmosis and coccidioidomycosis are two of those fungal infections that do tend to be on the severe side. They do tend to be systemic, along with that pneumocystis pneumonia that we talked about. The next type is subcutaneous mycoses. Sub-Q. This means subcutaneous means that it requires a break in the skin. Farmers tend to get subcutaneous mycosis infections a lot, or people that work in their gardens. They're working in their garden and they prick their finger on a thorn, and that is now an open wound, and there just happens to be certain type of fungal spores that now enter into that open wound. One example causes a disease known as sporotrichosis. Again, it's usually not fatal. It stays typically localized to the infected region. However, you do have to go to the doctor to get treatment for it because we don't have anything over the counter that's going to work as an antifungal. The third type are the cutaneous mycoses, also known as dermatomycoses or derma, because derma means skin. These are probably some of the more common types. These are the ones that we talked about a few minutes ago when I was talking about things like thrush and yeast infections and athlete's foot, and ringworm. These are all examples of different types of dermatomycosis because this affects the keratin-containing tissues, the hair, nail, and skin. Lastly are the superficial mycoses. I have a picture of this on the next slide. I really, really like the picture. I'll show you. But it tends to be localized on hair shafts and superficial skin cells. The thing about the superficial mycoses is they don't usually cause any problems, and in a lot of cases, people may have a superficial mycosis infection and not know it because they're completely asymptomatic, meaning they don't have any signs or symptoms of any kind of fungal disease. And really, you can only tell if a person has one of these superficial mycosis infections by literally taking strands of hair and looking at them under the microscope, looking for certain fungi. So again, they exist, but they typically don't cause any type of major problems. Opportunistic mycoses. These are a really big deal right now in the healthcare. Opportunistic infections are are becoming more common. So an opportunistic infection is basically where you have a pathogen that has an opportunity to cause a disease in a situation where it normally wouldn't cause a disease. For example, the two examples that I have on here, we talked about yeast infections. Well, for women that have strep throat, or if you go to the doctor and the doctor gives you antibiotics, a lot of times the doctor will also give you an antifungal at the same time. The reason for this is because when you take an antibiotic, that antibiotic is not specific to just bad bacteria. Antibiotics kill all bacteria, good and bad. Well, in the vagina, there is a normal level of bacteria and yeast, and the bacteria keep the yeast growth in check. Well, when you kill off all the bacteria, there is nothing keeping the yeast from growing very quickly and from flourishing, and before you know it, you have a yeast infection. So that's why a lot of doctors, like I said, will prescribe you an antifungal at the same time they're prescribing you an antibiotic. They'll tell you to take the antibiotic first, finish the antibiotic, and then take the antifungal. This is considered an opportunistic mycosis because if you hadn't had a disease or an infection that required an antibiotic, then you wouldn't have contracted the yeast infection. Okay, so it's almost like there has to be two diseases, disease A and disease B. You only contracted disease B because you had disease A. If you never contracted disease A, you never would have contracted disease B. So the other example here is pneumocystis pneumonia. That's that typically fatal type of pneumonia. I said it's very, very prevalent with HIV patients because they have a suppressed immune system. Well, if you're healthy, pneumocystis pneumonia doesn't really do anything to you. Your immune system is able to fight it off. Okay? So this is an opportunistic mycosis because basically you have to have disease A, which is HIV, in order to have disease B, which is pneumocystis pneumonia. So again, with opportunistic mycosis, there has to be two separate infections or two different things going on. And you have to have an opportunity for disease B to cause a disease because disease A existed first. Okay, so that is an opportunistic mycosis, and we'll be talking more about opportunistic infections in general later on as the semester progresses. Here's that picture of the superficial fungal infection that I said I would show. This is a human eyelash, and the human eyelash has a fungal infection. It looks like, just by looking at it, it looks like a yeast infection, some type of yeast that did not undergo the separation from the parent cell. So it looks like a chain of yeast cells. A lot of times, like I said, you would never know you had the disease or the illness unless you actually were plucking hairs and looking at them under the microscope to look for a fungal infection. Because like I said, a lot of times the person is completely asymptomatic. Okay, so continuing with our talk on fungal diseases, this is something, again, it has a very important role in history, and again, it's something that I find very interesting. Some fungi can stimulate allergies or produce something called mycotoxins. Again, that prefix myco means fungus. So anytime you see myco anything, so a mycotoxin is a toxin produced by some type of fungus. All right, so there is this fungus known as Claviceps purpurea. Okay. It produces what's known as the ergotoxin. This ergotoxin, or this particular species of fungus, tends to infect certain crops. In history, rye was a big crop that this particular fungus would infect. Back in the 16, 17, even 1800s, white bread didn't exist. The only bread that was available was brown bread because the brown bread was made from rye. Well, you see there's a witch's hat on this particular slide. So you may wonder why there's a witch's hat on this slide. And it goes back to the Salem witch trials. It was thought that the scientific explanation for these women supposedly having psychosis and being possessed by the devil or whatever, it was actually thought that they had ingested some rye bread that had the ergotoxin on there. And the ergotoxin, as you see, is a very, very potent neurotoxin. It can lead to seizures and psychosis and nausea, vomiting, and even death in the most severe cases. So this is likely what happened. There were a lot of women that were reported to have psychosis and hallucinations and things like that. And they think it goes back to the ergotoxin. Okay. So just a little bit of history thrown in there with some microbiology. Okay, so we are done talking about the fungi. Now we move on. We have one whole slide devoted to algae. Because to be honest, algae really aren't that interesting. Okay. So under algae, you see they are photoautotrophs. When we were talking about the fungi, I said they were chemoheterotrophs. The opposite of a chemoheterotroph is a photoautotroph. You can look at that word and break it down. Photo means sun, auto means self, troph is an organism. These are self-feeders. They use the sunlight to self-feed. They're able to make their own ATP just from using the sunlight. That is why they are called photoautotrophs. So basically anything that can perform photosynthesis is going to be a photoautotroph. Obviously, they are mostly found in oceans. There are two that I really want to talk about. The diatoms first. They are unicellular. They store energy in the form of oil. This is currently a topic of interest. The United States loves oil. Most countries love oil. They produce enough of this oil. There's some research going into this trying to figure out if they produce enough oil and if this oil can actually be used for something. So again, current area of environmental microbiology. But these diatoms, you can see a picture of a diatom in the upper right-hand corner. Some of them have a natural brown tint to them. Some of them are translucent or transparent. It really kind of depends on the environment that they're in. But they produce something called domoic acid. And it causes a neurological disease. Basically, from my understanding, it makes a person act drunk. The domoic acid, it makes them stutter, slur their words, they can't walk straight. So it's very much kind of like a drunk person. But the human body can break down domoic acid, but it does so very slowly. So a person may be in this drunken-like state for 2, 3, 4 days, depending on how much of the domoic acid they consumed. So these diatoms, they produce domoic acid, and usually, okay, I will say there have not been any human deaths related to domoic acid. No human deaths. But there have been other deaths, particularly when it comes to birds and certain animals that are in that area. Humans, when a human winds up with a domoic acid poisoning, it's because they ate mussels or oysters. And if you know anything about oysters, oysters are filter feeders. The purpose of an oyster is to take in water, dirty seawater. They filter out all the dirt and then they expel clean water. So an oyster is basically a concentrated sack of dirt from the ocean. Okay. So if that domoic acid, if that oyster feeds on a lot of diatoms in that area, or consumes a lot of that water, filters out a lot of that water, it's possible that the domoic acid will be in that body of the oyster for a long period of time in a large amount. And so if a human consumes that oyster, they can wind up with domoic acid poisoning. That's somewhat similar to the next one that we're going to talk about, the dinoflagellates. Now, these are cross-listed as plankton because the dinoflagellates have some characteristics of algae, but they also have some characteristics of the protozoa, and that's where plankton would fall into. These are more serious. They produce a neurotoxin which causes red tide. You can see a picture of red tide on this PowerPoint slide. So this is a much more serious condition. Humans have died from red tide poisoning. It also affects marine life, shrimp, oysters, things like that. Again, a lot of times a human will wind up with red tide poisoning because they eat oysters. And red tide is out, the oysters consume and they filter out the neurotoxin, and so then the person eats the oyster and then they wind up getting sick. We have had red tide a couple of times. off the coast of Georgia. The last time was a few years ago, and basically say the rule is after red tide goes away, because eventually it disperses through diffusion and it will just go away on its own. But the rule is you're not supposed to have any seafood. You're not supposed to eat any seafood for 4 months from the area where red tide was. Obviously, you're not supposed to go swimming or anything in that area either. Now, there was some very interesting news in the state of Florida a couple of years ago. I think it was actually right before COVID. The state of Florida issued an emergency statement basically saying that the entire state, on both sides of the state of Florida, were experiencing huge amounts of red tide. And because it's not safe when red tide is out, the state of Florida lost millions of tourism dollars because you can't harvest any type of shellfish from that area. You can't swim. A lot of, you can't go boating. You're not supposed to be in the area where red tide is. What was more interesting that I found was previous to this, it was thought that you had to be swimming in the area to contract red tide, or the neurotoxin that causes red tide. New research indicates from the state of Florida that there were people, and all they were doing was walking along the beach near the ocean where red tide was, and they contracted the neurotoxin. So now there is evidence that this neurotoxin is somewhat airborne, and you can contract it simply from inhaling it as you're walking along the beach. So like I said, that's one reason I love microbiology. It's always changing. We're always learning something new. Okay. All right, so we are done talking about the algae. Moving on to probably one of my favorite sections this entire semester are the protozoas. So the protozoas, remember that word breaks down into proto meaning first and zoa meaning animal, first animal. These are unicellular, therefore they are microscopic. They are eukaryotes, so for the most part, most of them are going to behave very much like a traditional eukaryotic cell. They are chemoheterotrophs, but it's a very, very large and diverse group. There's a few that are pathogenic. And I try to focus on the ones that are either very deadly worldwide or very common worldwide, or the ones that we really see here in the United States. Due to having proper sanitation and adequate health care, we don't have a huge protozoan infection problem like other areas of the world do. And so, again, we'll talk about just a couple where we have seen cases of certain ones here in the States. They're typically found in soil and water and as normal microbiota in animals. Most of them are swimmers, meaning they're going to have something to help them swim. They're going to have cilia or a flagella, or they're going to have a pseudopod. They're going to have something to help them move through water. Okay. Trophozoite. This is simply the active feeding and growing form. This is known as the vegetative form of the protozoa. It is actively feeding and growing. Now, how do they reproduce? So, again, this is where they do vary when compared to a traditional eukaryotic cell, because they have a variety of ways in which they can reproduce. They can reproduce through asexual methods, such as fission, which is very, very similar to binary fission in bacteria. Some of them are able to undergo budding, like we just talked about yeast budding. And then some are able to undergo schizogony. Now, schizogony is really cool. At least I think it's really cool. If you look at the picture at the upper right-hand corner, that is a picture of schizogony. You have one cell, or you start with one cell and one nucleus. Well, because the protozoans are unicellular, that one cell, that is the entire animal. Okay, so it has one nucleus because it's a eukaryotic cell. Multiple mitoses occur. So one nucleus will turn into two turns into four turns into 8. Again, it's exponential growth. And then at the very end, cytokinesis occurs, which is separation of the cytoplasm. And you wind up with 8 parasites or protozoa. So you started off with 1, and then several steps later, you have 8 offspring, or 8 total. You have the original parent and its 7 offspring. Okay. Now, that is really cool. For every one, it yields 7 new ones. Again, this is a reason why some parasitic infections get severe very quickly, because every parasite has the ability of certain species, of course, certain species of parasites, for every cell or every animal, it gives off 7 offspring every time it divides. It is asexual reproduction, so no exchange of genetic material. They should all be genetically identical unless mutation occurred. I always have to throw that in. And then some protozoans are able to perform sexual reproduction via conjugation, and we talked about conjugation when we talked about bacteria. Okay. Now, some protozoans produce a cyst that provides protection during adverse environmental conditions. So when a human winds up with a protozoan infection, and don't worry, we're going to talk about several of these protozoan infections. When a human winds up with one of these protozoan infections, well, if you were to swallow a protozoan, your stomach pH is about a 1. It's extremely acidic. Your stomach acid would kill the protozoan. However, if that protozoan is one of those species that produces a cyst, think of a cyst kind of like an endospore. It has a very tough protective layer around it. And so the cyst provides protection from the extremely acidic stomach pH. So when a human winds up with one of these protozoan infections, a lot of times it's because at some point there was a cyst and it was ingested. The goal for a lot of these protozoans is your intestines. And the reason being, we'll talk more about this when we get to the worms, the next section of this chapter. Your intestines is a perfect environment for a lot of these protozoans, as well as the worms that we'll talk about. And the reason being, think about it. They're chemoheterotrophs. They eat whatever you eat. So whatever you had for lunch that's in your stomach and passes to your intestines, they're able to get nutrients from that food in your intestines. The intestines is warm. It has a neutral pH. There's space, there's food, and there's no predators. So for the protozoans as well as the worms, they really love the intestines. It's the perfect environment. And so that's why they develop a cyst to get past those stomach acids so they can make it to the intestines. Okay, medically important protozoa. So here's a very short list of just some of the protozoas that I'm going to talk about, and then I'll talk about just a few of the distinguishing features, but then as we go further, the next few slides, we'll go back to each one of these and we'll talk about each one more in depth. Archozoas. Okay, so at the beginning of this chapter, we looked at the 3 domains, and Archozoa was on there. It was the first branch of the eukaryotic cell. So archozoas, even though they are eukaryotes, they lack mitochondria. Okay. Now, mitochondria, this is where ATP is made in eukaryotic cells. They still make ATP, even though they don't have mitochondria, but they make ATP more like bacteria do, and we'll talk about that. We have a whole chapter devoted to that. A couple of examples of the archozoas are the Trichomonas and Giardia. So Trichomonas vaginalis, which is sexually transmitted, and we'll also talk about Giardia, which can be found in contaminated drinking water. The next group are your microsporas. They're unique because they lack mitochondria. They also lack a Golgi. So if you go back to that 3-domain system and you find archozoas, if you go up one branch, you will find microsporas. So they lack mitochondria and they don't have a Golgi. If you remember what the Golgi does, I tell students it's like the mail center. It receives incoming packages from the nucleus, it separates everything, and it ships it out where it's supposed to go. Okay, so that's what the Golgi does. One thing to keep in mind is some of these parasites, some of these protozoans, have more than one portal of entry, meaning how did the protozoa get inside the body? Some are through ingestion, so your mouth. Some can enter directly in your eye. And that's the case with Microspora. If you were to swallow Microspora, it causes diarrhea and other typical GI problems. However, this is also one that can enter in directly through the eye, causing a condition known as keratoconjunctivitis. This is another one where there's a really strong correlation between AIDS patients because of their suppressed immune system and a particular protozoa causing keratoconjunctivitis. The third group are the amoebozoas. They're called amoebozoas. Again, your traditional amoeba are in this group, but they move via pseudopodia. We learned that word in chapter 1, pseudopodia. Pseudo means false, podia means foot, false foot. So they have these projections that stick out from them that they're able to move back and forth to help that particular parasite swim. And we'll talk about an amoeba, which causes dysentery. Again, that's just basically another name for severe stomach upset, severe diarrhea, severe nausea, stomach pain, things like that. And we'll talk about acanthamoeba, which is another one that causes dysentery, but it can also enter in other portals of entry. Apicomplexa, group 4. Apicomplexa is probably one of the largest groups, and it's probably one of the deadliest groups. And they are unique because they move by gliding. So Apicomplexa, they do not have flagella, they do not have cilia, they do not have pseudopods. They have no true means of swimming through any type of watery environment, but they are able to glide. They are also intracellular. In a lot of cases, it's red blood cells. You can actually look at a smear of red blood cells and you can see the parasite living inside the red blood cell. So those are pretty interesting. I said it's one of the deadliest groups, and it's one of the deadliest groups because of the first parasite mentioned, Plasmodium. This is the parasite that causes malaria. Okay, malaria is very, very deadly. Hundreds of thousands of people still die from malaria every year. Babesia, we'll talk about Toxoplasma and Cryptosporidium. And then the last group are the Euglenozoas. They're called Euglenozoas. They're also known as hemoflagellates. Heme meaning blood. Flagellates because if you look at the body of the protozoa, it literally looks like a giant flagella. And they're found not inside red blood cells, but they're found in blood smears. So again, that's why it's called hemoflagellates. We'll talk about two, Trypanosoma Trypanosoma cruzi and Trypanosoma brucei. And there was an outbreak in the state of Georgia about 2 or 3 years ago of one of those. Okay, so moving on to group 1, which are the archozoas. Remember, this is the group that lack the mitochondria. The term archozoa, they were named archozoas because they have features of the archaea and they have features that are zoa or animal-like. So the two that fall under this category that we're going to study is Trichomonas vaginalis and Giardia lamblia. With Trichomonas vaginalis, it causes trichomoniasis. It is considered to be sexually transmitted. Common symptoms include a strawberry cervix, which you can see over on the right, itching and smelly discharge. So if you look at the cervix is normally supposed to be like a solid pink color. They call it a strawberry cervix because it almost looks polka dotted. It is spotted with the dark pink spots. So as far as treatment, we do have treatment for Trichomonas. The most common treatment is probably Flagyl. It's an antibiotic, but it's also an antiparasitic. And if you look at the actual parasite itself, they're very fragile, very thin. They have flagella, again, for locomotion. They don't look like a traditional animal in any sense, but these are the protozoans, those single-celled animals. They are very small. They are microscopic. They have been shown to live on toilet seats and towels for 20 minutes. So there actually have been few, but or relatively few, but there have been a few cases where they've been transmitted to another person by the person sitting on a contaminated toilet seat. Moving on to Giardia lamblia, if you look at the parasite or the protozoa, it looks very similar to that of Trichomonas. It has multiple flagella for locomotion. Again, these are swimmers. A lot of these protozoans are swimmers. It causes giardiasis, which is commonly found in water. It causes what's known as traveler's diarrhea. So if you've ever traveled to another country and they tell you not to drink the water, this is most likely the reason. It's because their water has Giardia lamblia in it. It causes an extreme form of diarrhea and stomach pain, and other GI symptoms. Again, we do have treatment for it. It's a pill. But as you will see, based on there's a video that you will watch, there's a lot of people in the world that simply don't have adequate health care. They don't have clean drinking water. They don't have access to treatment. So if they were to contract something like Giardia, here in the United States, we usually see Giardia with people that are like backpackers and hikers, because, you know, they travel, they're hiking, they come upon some clear water. They think just because the water looks clear that it is clear, so they drink this water, and before long, they're doubled over in stomach pain. It can happen in as little as about 4 hours, but they're in extreme pain, and again, it's due to the parasite making its way to the intestines, releasing toxins, and causing the severe diarrhea. Okay, so moving on, group 2, these are microsporas. These are the ones that lack mitochondria as well as the Golgi. This is the one that has multiple portals of entry. It can cause diarrhea if it's swallowed. It can cause keratoconjunctivitis if it enters directly into the eye, but this is more with HIV/AIDS patients. It causes microsporidosis. There's over 1,200 species, but only 14 of those are known to infect humans. However, this is another one, just like pneumocystis pneumonia, where we're seeing a really strong correlation between HIV and this particular protozoan. So we are seeing an increase of the number of microspora cases. Now, symptoms include, well, it really depends on the portal of entry. If this is one of the protozoans that's swallowed, then you're going to have traditional GI problems, nausea, pain, diarrhea, things like that. However, if it enters directly into the eye, well, then you're going to have like foreign body sensation, pus, redness, irritation. So again, we do have treatment for it. We have antiparasitics. Ivermectin is a really common antiparasitic. It's a common, readily available, fairly cheap, again, as long as you live here in the United States or in other developed countries where you have access to medication. Moving on, really quick, just to look at the actual parasite itself, you can see it's the blue. It's kind of elongated. It's got multiple flagella on there because, again, it moves. It's able to swim or move. And you can see also, you can see the eye, and it's infected with the microspora, and it looks like pus, almost kind of like pink eye. The eye looks really irritated, and again, it's due to inflammation, due to microspora being present. Okay, the third group, Amoebozoa. These are named because they move via pseudopodia, false feet or false foot. So we'll talk about an amoeba and acanthamoeba, but it causes amoebiasis. The most common in humans is known as Entamoeba histolytica. This one is very serious. It causes numerous deaths every year worldwide. Again, really only affecting those underdeveloped countries that don't have access to medication. Symptoms start off on the milder side with diarrhea, stomach pain, and cramping. However, this is also one that triggers a fever. And this is one that can actually spread within the body. It can actually spread and move on to other organs. The liver is one of these. It can cause liver abscesses, and eventually what happens is the liver ruptures and the person dies of liver failure. So again, a lot of deaths from Entamoeba histolytica worldwide every year. Now there's acanthamoeba keratitis. Again, this is another eye infection. Also more serious because it can lead to permanent blindness, whereas the previous one, microspora, it was more of an irritation, foreign body sensation, but this one right here can actually lead to blindness because this is a parasite, again, that can enter into the eye. The third, granulomatomus amoebic encephalitis. Again, some of these amoeba have the ability to spread to different areas of the body, and this one has the ability to infiltrate sometimes the brain and the spinal cord, causing encephalitis. Encephalitis is a very, very severe condition that is often very deadly. Disseminated infection, that just means widespread infection, because like I just said, some of these parasites that are protozoas have the ability to infiltrate different organs, the sinuses, the lungs, the nose, respiratory. Okay, they can enter into the eye, they can invade the liver, they can, again, they can spread. Some of them are genitourinary, so they could potentially enter that way. So again, disseminated infection just kind of means widespread infection. I like the picture on the bottom right. This is an amoeba, and you can see the pseudopods. It's those long kind of projections, long skinny projections, and they are of different lengths and they are of different widths, but the amoeba has the ability to control the movement of the pseudopods to help propel that amoeba through some type of water, that environment. Okay, the deadliest group, group 4, is Apicomplexa. They move by gliding, so they don't have flagella or cilia or any means of movement. They don't have pseudopods. They move through gliding. They are also intracellular, so they reside inside cells. And a couple of these, such as Plasmodium, they like to be inside red blood cells. So let's talk about Plasmodium first. It is very, very deadly. This is the parasite that causes malaria. Now, there's multiple species. So Plasmodium is the genus. There's multiple species, but there's only 5 that have been found to infect humans. And it depends on which actual species you're infected with as to the severity of the symptoms and your chance of recovery. So the symptoms, again, they can be wide. Chills, fever, sweating, headache, malaise, which is basically just tiredness, muscular pains, vomiting, diarrhea. And like I was saying, it depends on the particular species. So right off the top of my head, you do not need to know this for the exam, but you have Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, and Plasmodium falciparum. And there's a 5th Plasmodium that always eludes me. I can't think of it. But if you were to be infected with something like Plasmodium ovale, you only have a 30% chance of death. 30% chance is still pretty high, but it's not as bad as Plasmodium falciparum. If you were to be infected with Plasmodium falciparum, you have a 90% death rate or chance of death. We do not have good cures for malaria. Okay, so the classic cure for malaria has been quinine, okay, which is taken from the bark of a tree. It's been used for hundreds and hundreds of years for other illnesses as well. Quinine tends to work for some of the lesser strains of Plasmodium, such as ovale and vivax, but it does not work for falciparum. So researchers, they tried to find another drug or a different drug, and they came up with the drug chloroquine. It's a derivative of quinine. It works a little bit better than quinine, but again, it does not work towards Plasmodium falciparum. They've also developed a third drug called quinolone. Again, it works for some of the weaker strains of or species of Plasmodium, but it does not work for falciparum. So the moral of the story is it really depends on the species you're infected with. Hopefully it would never be falciparum. Now, if you were to travel to a country where malaria is common, we do have medicines that you can take to prevent you from contracting malaria. But once you have malaria, the treatments are really sort of limited. So how do you contract malaria? Well, it's a parasite. It's carried in the mouth of a particular species of mosquito known as the Anopheles mosquito. and it has to be female. Only female mosquitoes bite humans. Male mosquitoes eat fruit. Female mosquitoes bite humans. And so the Plasmodium parasite is carried in the mouth of the Anopheles mosquito. And then the Anopheles mosquito, it bites a person and it deposits that Plasmodium parasite into that open wound. And then it gets into the bloodstream, it finds a red blood cell, and then it enters into the red blood cell. So the easiest way to determine if a person has malaria is simply doing a blood smear. Because you can take a finger prick, make a blood smear, and you can see the parasite actually living inside of the red blood cell. All right, so you see statistics in 2015, it says 438,000 people died of malaria. And that number really hasn't changed because, like I said, we haven't really made major advances in malaria lately, at any time lately. So roughly, I would imagine that many people are still dying every year from malaria. Moving on and talking about Babesia, it causes babesiosis. This is a lot more rare. Most students haven't heard of Babesia before, but it's transmitted by ticks. It is often misdiagnosed as Lyme's disease, but Babesia tends to happen more in the north, in the central region of the United States, in the colder regions, not as much in the south. So in the south, they automatically think, oh, Lyme's disease. But in the north, it could be Lyme's disease, but it could be Babesia. But like I said, it's often misdiagnosed. So you go to the doctor and they automatically think, oh, it's Lyme's disease because you maybe mention a tick or something. Well, the problem is they would give you antibiotics because Lyme's disease is caused by a bacteria known as Borrelia burgdorferi, and Babesia is caused by a parasite. So they give you antibiotic. Well, the antibiotic isn't going to work on the parasite, so you don't get better. You continue to stay sick until you get a proper diagnosis. Again, you can do a blood smear, and you can see the parasite living inside the blood smear or the red blood cell. Like I said, signs and symptoms are very, very similar to that of Lyme's disease. We do have treatment. Like I said, we have antiparasitics that work fairly well for Babesia, just not for Plasmodium. Toxoplasma gondii. This, in my opinion, is probably one of the most interesting parasites. Toxoplasma gondii causes a disease known as toxoplasmosis. In a lot of adults, it's completely asymptomatic. You have no signs or symptoms of it, and at some point it just kind of goes away. If you do have signs and symptoms of it, it's things like swollen lymph nodes, headache, fever, fatigue, muscle aches. It can last for about a month or so. But again, keep in mind, in a lot of people, it's completely asymptomatic, and that becomes important in a minute. The big problem with Toxoplasma gondii is with pregnant females. This is why, if you've ever been pregnant, at some point the doctor will ask you if you have cats, and if you have cats, to stay away from cat litter. Don't clean cat litter. That's because cats are what's known as the definitive host for Toxoplasma gondii. Toxoplasma gondii can live in a variety of different animals, but it can only reproduce inside of a cat. Therefore, it's always looking for a cat as a host so it can reproduce. All right, so cats are a definitive host. A lot of cats carry Toxoplasma gondii. It's passed in fecal matter, so that's why the doctors will tell you to stay away from cat litter pans if you are pregnant. The reason being, this is a parasite that, although is asymptomatic in most adults, if a woman is pregnant, it can cause major birth defects in the developing fetus. And I'm talking about missing limbs. The nose will be missing, an eye socket is missing, an ear is missing, major facial deformities. And so that is why, again, why doctors are always adamant about pregnancy and pregnant women staying away from cats. Now, there is new research. Okay, so this is where it gets kind of interesting. There is new research because, like I said, it was often thought to be asymptomatic in a lot of people. But there is new research that is now indicating that people infected with Toxoplasma gondii have a delayed reaction time. And they got this information by looking at car crash victims, at people that were driving cars when they crashed a car. And they looked at brain matter, and they looked at to look for this parasite because it can live in brain matter in humans. And they found that people's brains that were infected with Toxoplasma gondii had a delayed reaction time, and that was what was more responsible for their wreck. Also, they also found another piece of interesting research, and it's basically that Toxoplasma gondii, now they're saying it's associated with certain mental health disorders, namely schizophrenia and bipolar disorder. So people that have Toxoplasma gondii cysts on the brain are more likely to have schizophrenic-like symptoms or display some type of bipolar-like symptoms. So again, our knowledge of microbiology, again, is always changing. We're always learning something new. There's always new research being published. The last one that we're going to talk about is Cryptosporidium parvum. This is another waterborne parasite. It causes cryptosporidosis. It can be respiratory, but it's usually gastrointestinal. You have severe diarrhea, stomach pain, things of that nature. It ranks number 2 to Giardia lamblia. So if you go drink contaminated water, the number one most often the cause of getting sick is Giardia lamblia. If it is not Giardia lamblia, then it is most likely Cryptosporidium parvum. Cryptosporidium parvum ranks number 2. Remember, these are also intracellular. We're still in group number 4, which is Apicomplexa. So if you look at the picture at the bottom right, I really like this picture. It shows a cell rupturing, and it shows the parasite being released from that particular cell. A few years ago in Warner Robins, when Rigby's water park first opened, they were only open for a couple of months, and then they shut down for the entire year. And the reason they shut down was there was an outbreak of Cryptosporidium parvum. Now, Cryptosporidium is fecal oral, so what had to have happened was somebody was infected with Cryptosporidium parvum, and because it's fecal oral, I don't know if the person had diarrhea, but somehow Cryptosporidium was released into the water, and then that water splashed into the face of somebody else, and they contracted Cryptosporidium parvum. So again, it's fecal oral. So there were several cases of it. This is pretty common every summer. One year, Whitewater had to close due to an outbreak of Cryptosporidium parvum. Public swimming pools, every now and then I'll hear about in some state there was an outbreak of Cryptosporidium parvum at a public swimming pool. So again, this is a parasite that we see with contaminated water. Okay. And we do see this one in the United States. This isn't one that's strictly in other countries. We do see Toxoplasma gondii, and we see Cryptosporidium parvum here in the United States. The fifth group, these are the euglenozoas, also known as hemoflagellates. Because if you look at the blue structure, the picture at the top, it looks like a giant flagella. And it's called heme because it's found in a blood smear. It can be found in red blood cells. So that's where it gets its name, hemoflagellates. And there's one genus that's called Trypanosoma, but there's two species that we'll talk about, Bruci and Cruzi. Of the two, Bruci is by far the most deadliest. Trypanosoma bruci causes a fatal disease known as African sleeping sickness. And as the name implies, it really only occurs in Africa. I haven't heard of any cases like in the United States or anywhere else. There is treatment or a cure if the person has access to the cure. The symptoms start off mild, fever, headaches, fatigue, and it continues to get worse if left untreated. Eventually it causes neurological problems. The person will eventually slip into a coma and die, and it's usually a process. It's usually about a 6-month span from infection to death if left untreated. And a lot of people are still dying from African sleeping sickness every year. Next is Trypanosoma cruzi. It causes Chagas disease. This comes from the bite of a bug. There are a couple of different carriers or vectors, which we'll talk about, that can potentially carry Trypanosoma cruzi, one of them being the kissing bug. It's also known as the assassin bug. But basically it carries the parasite in its mouth, and then when it bites a person, it deposits the parasite into the fresh open wound. The assassin bug tends to bite on the face. It really likes the face region, the eyes and mouth in particular. You can see this person here. They have an eyelid that is inflamed. It almost looks like a sty, but it is not a sty. This is caused by the bite of one of these assassin bugs. The parasite has entered into the eye region. It's replicated, and it causes irritation, redness, but this is also one that can spread. If left untreated, it causes congestive heart failure. So again, it's a process. It's a long process, 6 months to a year. What's interesting about Chagas disease is we actually had an outbreak of it here in the state of Georgia a few years ago. It started off with a single case in Atlanta. There was a patient who was infected by something. The doctors tried, traditional antibiotics and antivirals, and nothing was working. The person was getting sicker. The doctor had no clue what the patient had. So they wound up taking a sample of the patient's blood, and it was sent to the CDC. It was the CDC that actually discovered what the patient had, that diagnosed Chagas disease. From there, it spread, and there were several cases of it in Atlanta, Georgia, and Rome, Georgia, where patients were coming down with Chagas disease. And so that is fairly rare for Georgia. It's fairly rare for the United States, but it's incredibly rare for the state of Georgia. So that's very, very interesting when you have something that even the doctors can't figure out, and so they have to send blood samples to the CDC. If the doctor had taken just a minute and done a blood smear, again, this is a parasite that will show up in the blood. So all they had to do is do a finger prick, do a blood smear, look at it under the microscope, and they probably would have seen the parasite. So yeah, that's the deal with Trypanosoma brucei versus Trypanosoma cruzi. Okay, moving on to the helminths. So the helminth, this is a term for a parasitic worm. Again, not all worms are parasites. So not all worms are considered to be helminths. Your earthworm is not a helminth. We are going to focus on just the helminths. They are in the kingdom Animalia because they are considered to be true animals. They are eukaryotes. They have traditional organelles. So we are going to be talking about the platyhelminths, which are the flatworms. Okay? And within this group, we're going to talk about the trematodes and the cestodes, or the flukes and the tapeworms. Flukes tend to be relatively short, maybe a couple of inches or shorter. The tapeworms can get 30 to 40 feet. And so we'll definitely talk about those as well. And we'll also spend some time talking about the phylum, the nematodes, which are the roundworms such as Ascaris and pinworms. There are very few of these that are considered to be human parasites. Humans are typically considered to be an accidental host. There is no worm called the human tapeworm. We have a beef tapeworm, we have a pork tapeworm, we have a fish tapeworm. There is no human tapeworm because, again, humans are an accidental host. We get in the way of what that worm was trying to do. We usually contract if we wind up with one of these parasitic worm infections. It's usually because we ate undercooked or improperly prepared food. They do have an anatomy and life cycle that's modified for parasitism. These helminths are considered to be true parasites. So when we were talking about relationships, these are a true parasitic relationship. They take whatever they need from the host, and they don't care what happens to the host. They don't care if the host survives or not. So if a human were to wind up with one of these helminthic infections, That helminth takes whatever it needs from its human host, and it doesn't care if the human host suffers or dies. Okay, so again, it fits the true definition of a parasite. But we'll talk about the anatomy on the next slide. They have very complex life cycles, and so because of that, we study them in microbiology because the majority of their life cycles, they're microscopic. Let's talk about the definitive host versus the intermediate host. So like I was saying, these worms or these helminths, they have very complex life cycles. There may be 12 to 15 stages in their life cycle before they become a true adult. Once they become a true adult, the adult stage is said to be the definitive host, or the adult stage can be found in the definitive host, whereas the different life cycle stages of that helminth can potentially live in different types of intermediate host, but eventually it will find its definitive host. So let me give you an example. Let's talk about the beef tapeworm. Okay. Well, as that beef tapeworm is going from egg through its multiple stages of life cycles, that beef tapeworm can live in a cow, it can live in a pig, it can live in a sheep, it can live in a human. Those are all considered to be intermediate hosts. Okay. Now, when that beef tapeworm, if it finds a cow and moves to that cow and lives inside that cow is now said to be the definitive host. So again, with the fish tapeworm, the fish tapeworm can live in a variety of different hosts. Any host other than a fish is said to be an intermediate host. Once it makes its way and finds a fish to live in, that is now said to be its definitive host. So that's the difference between an intermediate host and a definitive host. And if you look at that picture, she is standing next to a giant tapeworm. Yes, that is a tapeworm, one tapeworm. Because some of these tapeworms, particularly like the beef tapeworm, it can easily get 30 feet in length. Okay, two terms that you do need to know, and this is very, very kind of unusual. Monoecious versus dioecious, and this has to do with sexual reproduction. The flatworms are typically monoecious, and you will see this on the next slide. What monoecious means is that it contains both male and female reproductive structures within one worm. Okay, so that makes them very interesting. Again, this is only with the flatworms, such as the flukes and the tapeworms. They tend to be monoecious. They have both male and female reproductive parts. Dioecious is more what we're familiar with. This is where there are separate male worms and there's female worms. The roundworms, such as Ascaris and pinworms, they are dioecious. There are separate male and female worms. They are said to be more evolutionarily advanced than the monoecious worms are. So this is the grouping, the platyhelminths. And I said we would talk about the trematodes, which are the flukes, and the cestodes, which are the tapeworms. You can see a picture of a trematode or a fluke in the upper right-hand corner. These are considered to be worms. They max out at probably about 2 inches, but they can be much smaller. They are flat, and a lot of them are swimmers. Now, you don't typically think of a worm swimming, but these are classified as worms, and most of them are water-bound, meaning they are found in some type of water environment. Now, if you look, I said that these were considered to be monoecious. This one fluke has both eggs and testes. So one worm can have both male and female parts. That way they can act as male or they can act as female. And in some cases, they've been shown to be able to self-fertilize, meaning they don't have to find a mate at all. They can self-fertilize. They also have an oral sucker because, like I was saying, The goal for most of these worms is the intestines. We talked about why the intestines was such a great environment when we were talking about the protozoas. Well, once these worms make their way to the intestines, they need a way of staying there. Okay. And that is what the oral sucker does. They use the oral sucker to grab hold of the intestinal wall. Moving on to the tapeworm or the cestode, which you can see at the bottom, they're more complex. They can get 30, 40, 50 feet in length. I think the max length that one has ever been found was 60 feet, 66 feet, something like that. So there is the picture on the far right at the bottom. That is known as the scolex, which is the scientific name. That's actually the head region. So the entire thing is the scolex. And then the scolex has 4 suckers. You can see those suckers. And the purpose of the suckers is to, again, grab hold to the intestinal wall. They also have a rostellum. So the rostellum is in the very center. It's almost a beige tan color. And then you see the hooks around the rostellum. The purpose of the hooks is they bury the hooks into the intestinal wall. Again, it allows the worm to stay in the intestines. So they have hooks and suckers, and they use both of these to help grab hold of the intestinal wall. Now, if you look at the body of the tapeworm is broken down into individual segments known as proglottids. Okay. So each segment is a proglottid, and if you look at each proglottid has a testes on one side, an ovary on the other side, and a genital pore in the center. So each proglottid could potentially self-fertilize. Okay. Tapeworms that infect humans. The first one being Taenia solium. This is known as the the pork tapeworm. It can get 2 to 3 meters in length. So a meter is roughly a yard. So 3 meters is roughly 9 feet in length. They have 800 to 900 proglottids, 22 to 32 rotellar hooks. You prevent the pork tapeworm by either freezing the pork or by making sure the pork is thoroughly cooked. The picture of Taenia solium is the picture that's pink in the middle. You can see the suckers and you can see the rotellar hooks on it. Now the next tapeworm that we'll talk about is Taenia saginata. This is the beef tapeworm. It's 4 to 10 meters in length, so 10 meters, this is roughly 30 feet. But reports of 22 meters, this is roughly 66 feet in length, have been reported. They contain 1 to 2,000 proglottids, but they lack hooks. Again, you prevent the beef tapeworm infection by adequately cooking the food and by freezing the food, or through long periods of salting, which is lethal to the cysteria. So the cysteria is the cyst that some of these tapeworms can produce. There's a picture of the cyst on the bottom right-hand corner. It almost looks like an egg with a covering over it. That is a protective covering that's able to survive harsh living conditions. So to make their way past the stomach acids, to make their way to the intestines. A picture of the beef tapeworm. Remember, they lack hooks, but you can see a picture of the beef tapeworm on the top right picture. The suckers are in action. They are attempting to suck or grab hold, but there are no hooks. Next is Diphyllobrium. This is not one particular species. This is the genus, and there are multiple species within this particular genus. But this is known as the fish tapeworm. It can get up to 10 meters, which is roughly 30 feet. It has roughly 3,000 proglottids. It can release 1 million eggs per day, and it can live for up to 20 years. So again, very, very impressive. They can produce a lot of eggs. And then lastly is Hymenalepsis. Again, this is the genus, so there's several species within Hymenalepsis. This is known as the dwarf tapeworm. These are very, very small. You can actually see them without a microscope, but they are very, very small. And they do have the hooks, again, for or hooking on to the intestines. WHO, which is the World Health Organization, very important. They're headquartered in Washington, D.C. We will talk a little bit more about WHO later on. But again, the World Health Organization estimates that roughly half of the world's population is currently infected with some type of helminth. And here you can see it says that that is one tapeworm. Tapeworms reaching up to 60 feet long are the biggest parasite that live in humans. All right, moving on to the nematodes. These are the roundworms. Okay. These are considered to be more evolutionarily advanced because they have a complete digestive system, and there are separate male and female sexes. It's usually the eggs that are infective for humans. So once again, if you wind up with one of these infections, it's because at some point you swallowed one of the eggs. It made its way past the stomach acids and it made its way to the intestines. Ascaris lumbricoides. Okay, so Ascaris, it causes ascariasis. It's the second most common worm infection in the U.S. because it turns out that the next one we'll talk about, pinworms. They're number one here in the U.S. But Ascaris is the most common worm or helminthic infection in the world, with over 1 billion people currently infected. Now, Ascaris is very interesting because there's male and female sexes. They tend to hide in the intestinal tract. It is the females that are the most active. The females will come outside of the body at night, so they will exit the anus at night and they'll lay their eggs around the anus and then they travel back in the body. The reason they do this is to help spread the eggs onto a new host. You can see Ascaris has in the eye, the Ascaris entered the eye and it got embedded in the eye. And then you can see below that there's a bowl full of Ascaris worms that was taken from a person. Okay. Moving on to Anaerobius vermicularis, also known as the pinworms. This is the most common worm infection in the U.S. 30% of children have had pinworms. It is estimated that 16% of adults are currently infected with pinworms. Pinworms are fairly common. They're a very, very small worm. There is treatment for both Ascaris and for pinworms. Again, the females tend to come outside of the body. They leave the anus at night to lay eggs. That is why a telltale symptom of pinworms is, particularly in children, children will go around scratching their butt. And it's because the pinworms have come outside and they laid eggs, and those eggs are coated with enzymes that irritate the skin, which creates an itchy situation. So kids are prone to pinworms because the eggs are found in dirt. Kids go outside, they play in dirt, and they wind up with the eggs under their nails, and they put their hands in their mouth without thinking, and before they know it, the eggs have entered their mouth and they've made their way to the intestines, and the kid has a pinworm infection. So it's actually very common in children, even here in the United States. The heartworm, the scientific name is Dirofilaria immitis. This is the heartworm. It is transmitted by mosquito, by the bite of a mosquito. So this is the heartworm. It's carried in the mouth of a mosquito, and then the mosquito bites a person. It deposits the heartworm inside of a person. Now, heartworms are far more common in animals such as cats and dogs, but there have been human cases of heartworms. And as you see, heartworms, if left untreated, can be deadly. It causes congestive heart failure. And based on this picture, you should be able to see why. You have so many worms and those clumps of worms that it stops the heart from being able to pump blood properly. It stops the atria and the ventricles from being able to pump, and it stops those valves from being able to close properly. So you have blood leakage. And so eventually, if left untreated, whether it's an animal or whether it's a human, they will die of congestive heart failure if not treated. There is treatment, though, for both animals and humans. Okay, so we're getting near the end of the chapter. Arthropods as vectors. So an arthropod is basically an insect or an arachnid. We think of them, we typically just call them bugs. But to be a little more specific, they're called arthropods because they have an exoskeleton, jointed legs, and a segmented body. Now, within the arthropods, there's two classes. You have your insects, which are 6 legs, or which have 6 legs, and then you have your arachnids, which have 8 legs. A lot of times we'll typically use the term insect to talk about things like spiders and mites, but technically they are not in the class of insects. They are arachnids. So arthropods that carry diseases are known as vectors. We've talked about several different arthropods that carry diseases. We talked about Babesia and how ticks carry Babesia. So a tick is a vector. We talked about mosquitoes and how mosquitoes can transmit heartworms or malaria. So therefore, a mosquito is a vector. So again, a vector is any type of insect or arachnid that can carry a parasite from one host to another. It can carry a disease from one host to another. There has been a lot of research into the elimination of different diseases by eliminating the vector versus eliminating the parasite. If we look at malaria, for example, we have 2 choices. We could target Plasmodium, which is the parasite, or we could target the mosquito, or more specifically, the Anopheles mosquito. So we could target either one of those and it would knock out malaria. There would not be any more cases of malaria. It's really hard to kill something that you can't see. The Plasmodium parasite is so small that it's really hard to kill it. It's really hard to see it in order to kill it. However, a mosquito is really large. You can see a mosquito flying around. That's why there's been a lot of research and effort into, instead of killing the parasite, killing the vector that carries the parasite. Because if there's no vector, then there's no disease because the parasite can't get from host to host. Okay. Last slide. Here's just at the very bottom, there's just a couple of pictures of common vectors in addition to, like I was talking about, we have the assassin bug, we have mosquitoes, we have lice, rat fleas, deer fleas. Basically, they all bite. Okay. And let's talk about mechanical transmission versus biological transmission, because the vector doesn't necessarily have to bite. With mechanical transmission, say that you have a housefly, and houseflies don't bite, but you have a housefly and it just happens to land somewhere on a desk, and there happens to be a microscopic parasite right where the fly lands, and the parasite winds up on the fly's leg or the body of the wing or something like that, and then the fly flies off to a new location, it lands, and it deposits the parasite in a new location. That is considered mechanical transmission. The parasite is carried on the outside of the body of the vector. In this case, it was a housefly. With biological transmission, the vector has to bite because the parasite is carried in the mouth of the vector. The vector has to bite a host to deposit the parasite into the bite mark or the bite wound. So that is the difference between being a mechanical vector, between mechanical transmission and biological transmission. Biological transmission requires a bite, and mechanical transmission does not require a bite. And that is the end of chapter 4.